What's up, Ninja Nerds? In this video, we're going to be talking about bacterial genetics. But before we get started, if you guys really want to be able to understand this topic, down in the description box below, we'll have a link to our website. On our website, we have comprehensive notes. We have illustrations before everything's filled out on the board. And then we have all the illustrations after everything's filled out on the board. It's going to help you guys to truly understand this topic. So go check that out. Also, if you guys like this video, you benefit from it, please hit that like button, comment down in the comment section, and most importantly, subscribe. All right, let's get into it. All right, Ninja Nerds. So when we talk about bacterial genetics, the process by which bacteria actually replicate, it's a very simple process, right? You take one bacterial cell and make two bacterial cells, and then you just basically replicate their circular DNA. What is that process called? By which you take one bacteria prokaryotic cell and then replicate it into two prokaryotic or bacterial cells. We call that like mitosis, right? But there's a special term that we actually give that for bacteria, and it's called binary fission. Now this is a cool process because again, it's a way that we can, pass on the actual same genetic material or DNA from one cell to two cells. The only downside to this is that these DNA molecules are all the same. There's no differences, there's no genetic variability, there's no genetic diversity. So how do we diversify the actual DNA within bacteria? How do we do that? Well, there's four particular mechanisms. One is called bacterial conjugation. The second one is called bacterial transformation. We'll talk about all of these in great detail. The third one is called bacterial transduction. And last but not least is what's called bacterial transposition. Okay, so these are the ways by which we can diversify, right? So this is the ways by which we can diversify the bacterial DNA. Okay, that's important. So now let's talk about each one of these mechanisms. Let's go into detail about it, and let's talk about like the significance of those mechanisms. All right, so let's talk about conjugation. Conjugation is a very interesting way by which, again, bacteria can kind of change up the DNA from bacterial cell to bacterial cell. How does it do this? Well, one of the mechanisms we're going to talk about is this concept of fertility factor. It's a really cool concept. So let's say here I have a bacterial cell. This bacterial cell is what's called an F-positive bacterial cell, meaning it has the fertility factor. And I'll explain what that means. It means it has the ability to produce something called a sex pilus. This bacteria does not have the ability to produce a sex pilus right now, so therefore it's F negative. And again, the F is just standing for fertility factor, or the ability to produce a sex pilus. Now, how do I know what the heck all of this stuff is in the bacteria? Real simple here is this blue stuff here and this circular kind of structure here. This is our DNA, and this is like the chromosomal DNA. We'll just put like C for chromosomal DNA. This right here, this circular kind of maroon structure here, this is called a plasmid. And plasmid is basically like an extra chromosomal DNA. Okay? Now this plasmid has different genes on it that when transcribed and transmitted can make particular proteins. You know what one of those proteins are? It's proteins that make a tube that can branch between one bacterial cell to another bacterial cell. So you know what happens here is this plasmid. Let's say here's the plasmid, and on that plasmid there's a particular gene sequence, like right here. When that gene sequence gets transcribed and translated, it makes a particular protein tube. And that protein tube is called a sex pilus, right? So now let's actually show here a little tube connecting between this F positive cell and the F minus cell. So look, here's going to be a tube now that's connecting between these two bacterial cells, the F positive cell and the F minus cell. Okay, and again, the only thing that makes it F positive is the plasmid, which has the ability to have a gene to make the sex pilus. So that's the first step. So the first step here is basically utilizing the F positive cell, its plasmid, to make a sex pilus connecting the F positive cell to the F negative cell. That's the first step. What happens in the second step? The second step now is we should still have this pilus here that's connecting the F positive cell and the F negative cell, right? That should still be here. Here's our F positive cell, here's our F negative cell. Now, What's really interesting is that this plasmid, it's DNA. What can you do with DNA to make more DNA? You can replicate it. So what that actual F positive cell does is it replicates and makes another plasmid. So it takes the DNA here, here's our plasmid, and it replicates it. It makes a new plasmid. Okay? So it's going to undergo this process called DNA replication, where we take this plasmid, which has particular genes, maybe that gene is for actually making the sex pilus, and it's going to undergo that replication process. Now that we've made another plasmid and we have this little tube connecting the F positive cell to the F minus cell, what can I do? Pass that plasmid over to the F minus cell. Now, what's the result? So again, the second step here is actually replicating the DNA of the plasmid and then passing that plasmid over to the F minus cell. What do I have now over here? Well, I just replicated my plasmid, so I still have that plasmid that was the previous one. But now there should be a plasmid also present in this cell, and there was the sex pilus that was basically connecting them. Eventually this will start breaking down or depolymerizing. But now what do we have as a result of all of this? I have an F positive cell because it has that plasmid with the ability to produce a sex pilus. And what do I have now? I have another bacterial cell that at one point in time didn't have DNA that allowed for it to make a sex pilus, but now it does through this process of conjugation. Okay? That is an example there of that conjugative process. All right, so there's a similar concept with this conjugation. It's just a teensy bit different. There's another kind of conjugation process besides this sex pilus kind of passing on and replicating the plasmid DNA and passing that whole plasmid off to another bacterial cell. It's a little bit different. So this first step here, as let's say here, this first step, we have again, what kind of cells here? We have a bacterial cell, and this is the DNA, the chromosomal DNA, right? So this is the bacterial chromosomal DNA, and then this is the plasmid, which is the extra chromosomal DNA. This one doesn't have it, this one does. And let's say that that plasmid contains genes that can make proteins that can connect one cell to another via a sex pilus. That makes this F positive, and this one does not have that plasmid. It's therefore F minus, okay? Now what happens here is something really interesting. The plasmid says, hey, chromosomal DNA, I want to take some of my DNA and incorporate it into some of the chromosomal DNA. So that's what it does. The first step is we actually take, so imagine here we have this circular DNA, which is the plasmid, and then you have the chromosomal DNA. All I want to do is mix these two together. I want this plasmid DNA to be incorporated with inside of the actual chromosomal DNA. So as a result of that, you get something like this now. I have here my bacterial DNA, which is the chromosomal DNA particularly, and I have mixed into that the plasmid DNA. Now, something is very different now about this cell. This cell took its F plasmid, or the plasmid that contained the fertility factor on it, and incorporated it into the actual chromosomal DNA. You know what we call this bacterial cell now? We call this a high frequency recombinant cell. You're like, what the heck? That's what we call them. It's a high frequency recombinant cell. So it's basically a bacterial cell with chromosomal DNA and actual plasmid DNA that contains the fertility factor incorporated into the actual chromosomal DNA. That's a high frequency recombinant cell. This bacterial cell still doesn't have any kind of plasmid that contains the fertility factor, so it's just an F- cell. Now, here's the next thing that happens. In the second step of this conjugation mechanism, you have that plasmid DNA, right? And if you imagine here that we actually we drew this plasmid and chromosomal DNA combined. So here I have all of this DNA that's combined. On that plasmid DNA, which is incorporated into the chromosomal DNA, it has genes that can still make the sex pilus because it still has that plasmid there. If it decides to make proteins, that'll actually lead to a nice tube connecting the HFR cell, or the high frequency recombination cell, to the F- cell. It'll do that via the formation of a sex pilus via the genes on the plasmid that's incorporated into that DNA. Now we have this sex pilus connecting between the high frequency recombination cell, and the F- cell. Then what happens is, here's where it gets really interesting. I'm going to take some of the actual DNA from the plasmid and some of the DNA from the bacterial chromosome and actually transfer some of that over via the sex pills. I'm not going to transfer all the entire plasmid DNA. I'm going to transfer some of the plasmid DNA and some of the actual chromosomal DNA. And so as a result, some of that actually gets passed over. And you get something like this that gets passed over. You get maybe like a little piece of bacterial DNA and a little piece of plasmid DNA that gets crossed over via the sex pills, not the entire plasmid. Now, as a result, after you pass over that kind of mixture of plasmid DNA and bacterial DNA, what's the result out of this? Okay, well, you're still going to have a high frequency recombinant cell here. So it's still going to be a high frequency recombinant cell. And you're still going to have an F- cell because this F- cell didn't get all of the plasmid, and therefore it actually didn't get the genes necessary to be able to make a sex pill. So therefore it's F-. But it did get some of the plasmid DNA, and it did get some of the bacterial DNA. So it also has some of that plasmid DNA and some of the bacterial DNA. But it doesn't have the portion of the plasmid DNA that allows for it to be able to make the sex pilus. So therefore it is an F- cell, but it has some differences in its DNA from previously. So it underwent recombination, but not high frequency. So we call this a recombinant F- cell. That is another example of the conjugation process. You're probably like, why do I need to know all of this stuff? What's the purpose of this, Zach? Don't worry, I gotcha. What is really, really cool about this with the plasmids is not only their ability to make a sex pilus, right? So one of the concepts here is that they can make that sex pilus. But you know, on that plasmid, something very interesting. It may contain a very specific type of gene. So say that I zoom in on that plasmid. And let's say that we have this plasmid and it has a very particular gene. And this gene, when it's transcribed, makes kind of an RNA, and then it gets translated and makes a protein. And this protein that it makes is very interesting, and it's called a beta-lactamase. You know what beta-lactamase does? Beta-lactamase basically inhibits, you have like, for example, penicillin. Penicillin is a beta-lactam antibiotic. It has a beta-lactam ring within it. If you take penicillin and it interacts with this beta-lactamase, it basically breaks down and inhibits penicillin's efficacy, and therefore won't be able to exert its antibiotic effects on that bacteria, right? That's one of the interesting things about this plasmid. So if I took bacteria and I put them into let's say a plate, and I put some penicillin on that plate, this bacteria, if it has the plasmid that contains that gene, it would survive despite the penicillin being there. But this one, would it? No. So what if this conjugation process allows for me to take a bacteria that's nearby on like an actual kind of like a plate, like a, let's say a petri dish, and I put these bacteria in the actual petri dish and I put some penicillin in that area. If possible, this bacteria, which has that plasmid to produce the beta-lactamase, may make a sex pilus and pass that actual plasmid DNA over. Now this bacteria may have the plasmid DNA to make the beta-lactamase and help it become resistant to the penicillin antibiotics. So this is an interesting concept where conjugation can become involved in antibiotic resistance and passing on genetic material from one cell to another, enhancing their evolution and adaptation process. All right, beautiful. Let's talk about transformation. All right, so the next type of bacterial genetic mechanism that we got to talk about is transformation. So transformation is actually a very simple process. So it's the process by which we take DNA from kind of basically the extracellular environment and take it into a bacterial cell and then incorporate it into that bacterial DNA. So let's say that we take, for example, here we have a bacterial cell, and let's kind of term this bacterial cell a harmful, let's call this as a harmful type of bacterial cell, right? And let's say here And this bacterial cell, which has the blue DNA, is harmless. So in other words, it doesn't really have the ability to cause like a lot of damage to the host cell or tissue cells. If I put this, like for example, let's say it's in the body, or let's actually use that example. Let's say it's in the body, this bacterial cell gets into the person's body, and it actually, the immune system is able to destroy some of those bacterial cells. It destroys some of these actual bacterial cells, these harmful ones. When they're destroyed, they can release out some of their actual chromosomal DNA. And some of it may get broken down and fragmented. But the result of that is that out here in the extracellular space, you have some of this actual chromosomal DNA from the harmful bacteria. This harmless bacteria, which is in close proximity, if it is competent, so if it is a harmless bacteria that is competent, meaning it's capable of taking up this actual extracellular DNA from another bacterial cell, it will then take up this actual, we'll call this naked DNA. We're going to call this kind of the naked DNA from the harmful bacteria. It's going to get taken up into this bacterial cell. When it gets taken up into this bacterial cell, some of that actual naked DNA, parts of it, may be incorporated into the DNA of this other bacteria. So what would that look like as a result? As a result, you may have something like this. Now I have a bacteria, which is basically, this was previously kind of a harmless bacteria. We're going to use that term, harmless bacteria, with what now? DNA, with DNA from harmful bacteria. You're like, why is this important, Zach? Don't worry, guys, I gotcha. Okay, you know what the problem is that harmful bacteria, they're not harmful just because that's their natural innate ability. It's based upon the actual DNA itself. The DNA is able to be transcribed, translated, and then make proteins, and those proteins are able to cause negative, damaging types of effects on different types of host cells. Well, guess what? I picked up some of the DNA from that harmful bacteria and I just incorporated into this actual DNA of the harmless bacteria. It may have the ability to make proteins now that are damaging and nasty in nature. So now, as a result of this, let's say that we take this bacteria now. We have this bacteria, it has its original DNA, but now a part of it is the DNA from the harmful bacteria. Now, that harmful kind of like DNA that we can say, it may have genes. And those genes, when transcribed to make RNA, is then translated and makes proteins. And these proteins that they make can be really nasty. They can produce devastating types of effects. So they may produce very harmful types of proteins. And some of these proteins may be exotoxins. And that is the big thing to remember. Okay, what I want you guys to take away from this is what type of bacteria, because this is definitely going to show up on your exam, what type of bacteria have the ability to produce this transformation process, allowing for them to become competent, take up DNA, and then make nasty proteins as a response from that. All right, so what is the significance of this transformation process? Well, you have to remember there is particular types of bacteria, and you will get asked this on your exam, that have the ability to do this transformation process and make harmful types of proteins or exotoxins that can cause devastating effects. And it can really kick you in the shin. You know, that's actually kind of a helpful mnemonic, and I'll show you how that mnemonic works. So you have three particular types of bacteria. The S in the shin is Streptococcus pneumoniae. The HI in the shin is Haemophilus influenza, and particularly type B. And the third one is Neisseria meningitidis. These are the three particular types of bacteria that you want to remember perform this type of transformation process. Let's now move on to transduction. All right, the next type of mechanisms by which we can actually diversify or kind of cause changes within the bacterial DNA is this process called transduction. And there's two types, generalized and specialized. And we'll give you examples for a very, very high yield to remember for your exams for specialized transduction. All right, so how does this process work? Transduction is basically a way that we can transfer or change up or diversify DNA through the kind of a carrier or vector, and this is via a bacteriophage. So we had conjugation, we passed it on through a pilus. We had transformation, as we had a bacterial cell take up the DNA extracellularly. And then the third one, which is transduction, is we have some type of kind of vehicle or vector that's going between bacterial cell to bacterial cell via a bacteriophage. Okay. So generalized transduction. Let's say here I have a bacteriophage. A bacteriophage is basically, this is just kind of a virus, and inside of it, it has DNA. Okay, so this is what we call a bacteriophage. So it's basically just a virus with its own DNA that likes to infect other bacteria and then basically help to make more bacteriophages and pass on genetic material. Well, I'll explain what all that means. Okay, ready? Here we have this bacteriophage. Let's say it binds onto this bacteria, right? So it binds on, latches onto this bacterial cell. And when it latches onto this bacterial cell, it injects in some of its actual DNA, which is like a viral DNA, but we like to call this phage DNA. It's just viral DNA. So here I inject in that viral DNA. What is it again? It's viral DNA, but sometimes we also use the term phage DNA. Okay? Now, that's the first step. The first step is I use a bacteriophage which has viral DNA or phage DNA and have it bind onto a bacteria and inject that actual phage DNA in. Once the phage DNA is inside of this bacterial cell, it uses particular types of enzymes and proteins, and basically it'll replicate and make more phage DNA. It'll also make particular proteins and enzymes that'll actually help to break down and segment apart some of the actual bacterial DNA, and it'll also utilize some of its genes to make particular types of proteins that'll be helpful in replicating some of those actual viral particles, or in this case, the bacteriophages. So three things as a result of this happen. One is you actually use that phage DNA or viral DNA to make proteins that will enable it to make more of these bacteriophages. And the third one is release enzymes that break down and segment apart some of the bacterial DNA. So what does it look like afterwards from that effect? So the effect of this is you're going to have in different parts, in different areas of this bacterial cell, you'll have some of these kind of proteins which we call capsids. They'll be surrounding what things? Well, some of the capsids will surround some of the phage DNA or viral DNA, and some of the particles, some of these capsid particles, will be surrounding some of the bacterial DNA. But either way, I have some of that actual DNA here of bacterial DNA involved within these capsid proteins and some of the actual phage DNA incorporated within these capsid proteins. As a result, this is just going to continue to keep happening, and eventually you're going to have something like this. Eventually you're going to have so many different types of bacteriophages that are being made within inside of this bacterial cell. There's just going to be tons of them. And they're going to keep continuing to replicate and replicate and replicate. And again, some of them are going to have phage DNA, and some of them are going to have the bacterial DNA from that original bacteria. What's going to happen is you're going to accumulate so many of these viral particles that eventually it's going to lead to the lysis, or the popping of this bacterial cell. When it pops, it starts releasing out these phage particles, these bacteriophages. Some of the bacteriophages, again, contain what? Phage DNA or viral DNA, and some of them contain bacterial DNA. Which one do you think I actually really care about? I actually care about the bacterial DNA, because that's the one that I want to pass from this bacterial cell to this other bacterial cell. So now look, I pop this little guy out. I pop out that bacteriophage. Now inside of this bacteriophage, I'm going to have some bacterial DNA from this other bacterial cell. We'll call that bacterial cell number 1. This is the bacterial cell number 2. I'm going to use this bacteriophage and inject in what? The actual bacterial DNA. That bacterial DNA now is going to do what? It is then going to get incorporated into this actual bacterial DNA here. And from that, if that happens, I'm now going to have, as a result of this, a bacteria which contains its own bacterial DNA and a mixture of bacterial DNA from another type of bacterial cell, making it different, giving it variability, giving it genetic diversity, allowing for it to maybe have genes there that allow for it to make proteins that could be potentially harmful or make it resistant in some way, shape, or form. So it's a really cool process by which this generalized transduction can occur. All right, so let's talk about the second type of transduction, specialized transduction. Very cool process, almost the same as generalized for the most part. There's just a couple idiosyncratic details that we have to differentiate. Okay, so here we have our bacteriophage. Same thing as before. The bacteriophage is basically a virus that contains phage DNA, and it just likes to infect other bacteria. It transmits DNA from bacteria to bacteria. So this is our bacteriophage. And inside of that bacteriophage, we have that kind of maroon colored DNA. That is phage DNA or viral DNA. It's going to bind to the bacteria and inject that in. That's the first step. So it binds and injects the actual viral DNA or phage DNA into the bacteria. The second step is that phage DNA or viral DNA that we inject in, remember how in generalized it actually would get transcribed, translated, make proteins, enzymes that break down and segment the DNA? It would then also replicate itself, and it would also make capsid proteins. Well, what this one does is it takes and gets directly incorporated right into the bacterial DNA. Straight in it goes, baby. And as a result of that, within this next kind of view here, we're gonna have this bacterial DNA, and incorporated into it is going to be the phage DNA or the viral DNA. And you know what's going to happen is that phage DNA or viral DNA is actually going to start utilizing some of the genes to make capsid proteins. So I'm going to start getting tons and tons of capsid proteins that I'm also going to produce. And another thing that's going to happen is that this actual DNA, all of it will start kind of undergoing replication. And I'm going to start replicating some of this actual DNA. And the DNA is going to be a mixture of bacterial DNA and phage DNA. Then what I'm going to do is I'm going to start incorporating all of these things together into the capsids. So as a result, what will this look like in this next bacterial cell picture here? So here, I'm going to have tons of capsid proteins that I synthesize from that phage DNA. And these are going to be kind of taking up most of the host cell, or this bacterial cell. Inside of it, I'm going to have DNA, bacterial DNA, and I'm also going to have a little strip of phage DNA incorporated into it. Then what's going to happen is this bacterial cell, after it occupies all of this actual bacteriophages, is eventually going to undergo lysis and pop. And it's going to release out some of these bacteriophages that contain capsids, and inside of the capsids it has a mixture of bacterial DNA and phage or viral DNA. Then that bacteriophage, which was actually going to be, again, kind of formed from infecting the bacterial cell number 1, is going to go and infect the bacterial cell number 2 with different DNA. But what is it going to inject into it? It's going to inject into it DNA that is bacterial DNA from bacterial cell number 1 and phage or viral DNA from that original bacteriophage. And that's going to get put into this bacteria. So here comes this DNA, which is kind of a mixture of bacterial DNA and phage DNA. And guess what? It's going to get incorporated into the DNA of this bacterial cell number 2. And as a result, you're going to get a huge change in diversity here. You see how this is very specialized. So now I have a mixture of original bacterial DNA, some different bacterial DNA, and phage or viral DNA within this last cell. That's a huge change. So there's a lot of genetic diversity and variation from bacterial cell to bacterial cell via this transduction process. Now, the significance of this is particularly that certain bacteria perform specialized transduction, and it enables them to produce very nasty exotoxins. And you can remember the mnemonic by A, B, C, Ds. So A is for group A, Streptococcus pyogenes, which produces what's called a erythrogenic toxin. Okay, so this is a type of exotoxin. B is for the botulinum, so the botulinum toxin. C is for the cholera toxin. D is for the diphtherial toxin. And S is for the Shiga toxin, which is produced by Shigella. Diphtheria is produced by the Corneobacterium diphtheriae. Cholera toxin is produced by the Vibrio cholera, and then botulinum toxin is produced by the Clostridium botulinum. And then again, the erythrogenic toxin is produced by group A, Streptococcus pyogenes. If you really wanted to remember that, it's produced by what's called Streptococcus pyogenes bacteria. So that's what we call group A, Streptococcus pyogenes, who produces the erythrogenic toxin. That covers that. Let's finish off the bacterial genetics with transposition. All right, so we're on the last type here of the bacterial genetics discussion, which is transposition. Now, transposition is really cool, and it really comes into play with, like, drug resistance against certain types of bacteria. All right, so how does transposition work? Let's say I have, again, my chromosomal DNA. So this is my bacterial chromosomal DNA. And then here's my plasmid, right, which is, again, an extra chromosomal DNA. Let's say that on my chromosomal DNA, I have a particular portion here that I'm going to zoom in on and look at. So I have a particular portion of the chromosomal DNA that I'm going to zoom on and really, really look at, because it has a very particular type of gene sequence or element that I want to talk about. This element, which we're zooming in on, is what's called a transposable element. A transposable element. It has two particular parts. So it has this thing called your insertion sequence, which is this purple part. And then on the sides here, it has these things called inverted repeats. Okay? And what I really want you guys to remember about this is that on this sequence of DNA, which is going to be a part of the chromosomal DNA, it has this thing called a transposable element, which is basically a gene. And this gene, when transcribed and translated, makes a very particular type of protein called a transposon. So it makes something called a transposon. This transposon is an enzyme, and it's a very interesting enzyme. What this enzyme does is it does two things. It comes back, identifies the actual transposable element where the insertion sequence is and the inverted repeats are, and it makes these cuts within that area just outside of the inverted repeats on opposite ends, on opposite parts of the strand. Okay? So it makes these cuts like right here. So that's what the transposon will do. It'll be synthesized from the transposable element, go back and recognize the site from which it was actually transcribed, and cut on the sides of the inverted repeats on the opposite ends, okay, of the opposite DNA strands. Then what it does is it finds a particular sequence or a target sequence on DNA maybe downstream from this chromosomal DNA, or a part of extra chromosomal DNA, like a plasmid DNA. So basically, this DNA could be of two parts. It could be still a part of that chromosomal DNA, or it could be a part of the plasmid DNA. Okay, this one we're just particularly zooming in on is just from the chromosomal DNA. But again, what this transposon is it's a protein or enzyme made from the transposable element on the chromosomal DNA. It then identifies the actual transposable element that it was made from, makes nicks on the side of the inverted repeats on opposite ends. Then it identifies a target sequence. So this is going to be what's called our target sequence in pink here. So this is our target sequence. And this target sequence that we have here in pink could be just downstream from the chromosomal DNA, or it could be on the plasmid DNA. Then what it does is it takes the DNA from these portions here of the actual chromosomal DNA and inserts it into the center. So it makes like a little cut or a nick here right down the middle of the target sequence. When it makes that cut, it then inserts in this insertion sequence with the inverted repeats right into the center of it. So what does that look like as a result of it? As a result, I'll have the target sequence right here on the sides, because I kind of made like a little cut in the center of it, right? So there's my target sequences. Then I'm going to insert in the actual transposable element. And on the sides, what were there in maroon? the inverted repeats. So here's my inverted repeats. And then right in the center is what? My transposable element. Well, the insertion sequence part of it, right? And that's going to go right here in the center. And this is now my new DNA that I have that's either incorporated into a plasmid or it's incorporated into the actual chromosomal DNA downstream. Let's say, for example, let's use the example that this is one of the two. It's either a new part of the DNA, which is in the plasmid, or it's within inside of the other part of the chromosomal DNA. Let's just use plasmid as an example to explain the significance of why this is important. Let's say that this gene that we're talking about here, that we're actually going to try to transpose, is a very interesting gene that's important for antibiotic resistance. So let's take an example here. Here we have a bacteria, right? And we're going to call this bacteria an enterococcus, right? An enterococcus. And let's say that it has kind of this very important gene that we want to focus on. Here's that gene. Okay, so here's the DNA. And here's that particular gene that we're going to focus on. This gene is called what's called a Van A gene. It's called a Van A gene. The Van A gene is a gene that whenever it gets transcribed and translated, it makes proteins, and these proteins that are actually synthesized alter the structure. So they alter the peptidoglycan layer within the bacteria. Okay? So again, you have this enterococcus. It has a very particular gene that we want to transpose to another bacteria, and that gene is called the Van A gene. That Van A gene basically, whenever it gets transcribed, translated, it makes proteins, and these proteins basically alter the structure of the peptidoglycan layer within the bacterial cell wall. What that does is it alters it in such a way that it leads to, you know, there's an antibiotic called vancomycin. The vancomycin is no longer able to cause kind of destruction or alter, basically destroy the actual cell wall, the peptidoglycan layer. So because this Van A gene leads to changes in the peptidoglycan layer, it leads to vancomycin resistance. Okay? And now that actual bacteria will be able to survive even in the presence of vancomycin, which is a very powerful antibiotic. And this can lead to something called enterococcus. You can have what's called vancomycin resistant enterococcus. So you can have what's called vancomycin resistant enterococcus type of bacteria, which is basically bacteria like enterococcus that are resistant to vancomycin via this Van A gene. Now, what if I wanted to take that Van A gene and pass it on to another type of bacteria? Maybe I wanted this actual gene to get incorporated into the DNA of Staphylococcus aureus' DNA. But I had to do that by maybe passing on that bacteria via conjugation, transformation, or maybe through the transduction process. And then once that DNA is into this actual other bacterial cell, we can then utilize this transposition process to incorporate some of that actual DNA, the Van A gene, into the DNA of the Staphylococcus aureus. Right? So now what happens is, let's say over here, you take that enterococcus, let's say here's our enterococcus, and what it does is, by some process, it's able to pass on some of its actual DNA. And on that DNA, you contain that Van A gene. And the way by which this is taken into the Staphylococcus aureus could be by the conjugation via Sax pilus. It could be via some type of transformation where it's competent enough to take it up. Or there could be a bacteriophage that's taking that DNA and injecting it into this bacteria. Either way, it's getting that DNA. Once it's then there, it's able to incorporate it or transpose it into its DNA. And then from there, now the Staphylococcus aureus has what kind of gene? It has the Van A gene. And now that it has that Van A gene, it then can do what? It can alter the peptidoglycan layer of its cell wall. It can lead to vancomycin resistance. So this Staphylococcus aureus can survive even in the presence of vancomycin. And then that leads to what's called vancomycin-resistant Staphylococcus aureus. So that's one of these really interesting things that transposition can do, is it can lead to resistance to antibiotics by causing the transposition of particular genes or DNA sequences from one type of bacteria to another type of bacteria, and leading to antibiotic resistance, in this example, the vancomycin resistance via the VAN-A gene. All right, engineers, in this video we talk about bacterial genetics. I hope it made sense. I hope that you guys enjoyed it, and as always, until next time.